A major area of our lab focuses on understanding the role of regulatory non-coding RNAs in oxidative stress responses. There has been an increasing interest in understanding how environmental stresses affect human health, specifically at the molecular level when it comes to changes in gene expression. At the molecular level, there are a number of environmental inputs that have been well documented to augment the number of already existing endogenous reactive oxygen species inside cells. This raises the natural concentration of species like nitrogen oxides, peroxides, hydroxides, etc., that cause a number of damages in proteins, lipids, and nucleic acids. For proteins, these damages can include structural and functional modifications. For nucleic acids, they can include strand breaks, crosslinks, and chemical atoms. Importantly, these chemical changes take place despite native antioxidant defenses present in the cell. Given the major focus that has been placed on understanding oxidations on DNA and proteins, in our lab, our focus has been specifically on RNAs. Our interests have been focused on especially non-coding RNAs, as these are known to play key regulatory roles throughout all domains of life. To clarify, these are typically not translated RNAs, with a few known exceptions that encode for short peptides. So a key fundamental questions that we have been working on is, what role do RNAs play in regulating oxidative stress responses? Mechanistically, it is possible that RNAs are highly sensitive to oxidative stresses, given reports that these molecules are oxidized 14 to 25 times more than even DNA. For instance, oxidation at the guanosine, shown in this cartoon, the so-called 8-oxoguanosine or 8-oxo-G oxidation, is a well-characterized modification on RNAs that we sought out to track as a marker of stress. In toxicology, oxidative RNA marks have not been traditionally used. Instead, two protein markers, IL-8, which measures inflammation response, and lactate dehydrogenase, LDH, which measures cell death, have been traditionally used. More recently, microRNAs, especially microRNA 10B, involved in stress response, cancer signaling, and inflammation, have been used as stress response indicators. To start exploring how RNA molecules could be used as markers of oxidative stress, we have been exploring the accumulation of RNA oxidation marks, or 8OG, in a model of human lung cells exposed to air toxins. Specifically, we have been exposing B2B and A549 lung cells to defined air pollution mixtures containing ozones, acrylanes, and methacrylanes that lead to even more complex oxidative chemistries. It should be noted that the levels of these compounds in an initial studies, although relatively high for proof of principle, are relevant to situations with high air toxins and low diffusion variance, almost like standing in traffic between two tall buildings. Additionally, the ability to include UV radiation for sunlight generates high concentrations of radicals and highly reactive mixtures of toxins. One of our first findings in this area has been that RNA guanosine oxidation is significantly and more consistently increased in exposed cells relative to IL-8 and LDH markers. In this slide, we show cells exposed to mixtures of carbonyl alkanes and ozones through the black bars, and the white bars show controlled non-exposed cells. Importantly, among the two independent biological replicates we conducted, only 8OG increases were significantly detected in all experiments. Inspired by our lung cell work, we have also been looking at RNA changes during oxidative stress responses in a simpler bacterial system using D.radiodurans. This is an extremophile which represents a remarkable model for oxidative stress responses. For instance, one of its unique phenotypes is to survive extreme levels of ionizing radiation. Shown in this graph, it's its high survivability even at acute exposures of 10 to 15 kilograys of ionizing radiation. This represents 3,000 times higher survivability than human cells under the same conditions, and much higher survivability than E. coli cells, which can survive up to roughly 3 kilograys of ionizing radiation. So our lab has been working towards discovery of non-coding regulatory RNAs that are particularly associated with oxidative stresses. For this reason, we have developed a number of experimental and computational pipelines involving omics experiments and bioinformatic methods. An early result we have obtained is identification of a high number of non-coding RNAs observed uniquely in response to oxidative stress, specifically during recovery from high levels of ionizing radiation. In the slide, we represent some of the non-coding RNAs that we have recently discovered in D.radiodurans. Most interestingly, as shown by the non-coding RNAs boxed in red, some of these drastically, naturally change expression levels during recovery from ionizing radiation. It is important to review the suspected function of these intragenic non-coding RNAs at the molecular level. At the molecular level, these bacterial non-coding RNAs can perform a number of functions by targeting cellular mRNAs. As shown by panel A, they can downregulate mRNA expression by blocking the ribosome binding site, here shown as a Schindel-Garno. As shown in panel B, they can upregulate mRNA expression by increasing stability of the transcript mRNA or by competing with factors that can cause early termination, like the Rho factor in E. coli. As shown in panel C, regulatory non-coding RNAs can also downregulate expression by destabilizing the mRNA directly or by causing its degradation. In addition to our focus of intragenic non-coding RNAs that work by independently being transcribed and targeting mRNAs, we have also expanded our experimental and computational searches to understand the roles of non-coding RNAs found in untranslated regions within mRNAs. Indeed, we have recently discovered a highly structured 5' untranslated region within the gyrase gene. The gyrase gene encodes for the gyrase subunit A, which is a critical part of DNA repair during recovery from ionizing radiation. This 5' UTR regulates gene expression of its nearby gene gyrase in response to differential levels of radiation. As shown by the graph and the fluorescence image on this slide, we see more gene expression as ionizing radiation levels increase using the 5' UTR in the context of a synthetic GFP reporter. Moreover, this 5' UTR seems important for cell survival, as deleting it leads to a growth defect as observed in the lower right panel by the green curve relative to the other curves in which the UTR is still present in the. In conclusion, what we have shown is that RNAs can be highly sensitive to oxidative stress through changes in chemical modifications. We've also shown that non-coding RNAs are present in non-model organisms that can naturally go changes in expression levels under oxidative stresses. Lastly, we have also discovered non-coding RNAs found locally at 5' UTR regions of key genes in critical pathways that contribute to cell survival and recovery under ionizing radiation. These represent additional layers of regulation to known radiation responses in this organism that we are currently investigating.